Hello students, welcome to Raaonline. Our today's topic of crash series, biological classification. Biological classification, actually the number of organisms, number of different types of species in this world which are discovered so far 1.7 to 1.8 millions. This is the number of species which are already discovered, described as well as named. Such a large number of species is there, so one by one study is not possible. In that case, the various attempts of classification were made in the past also to classify them on the basis of their similarity and differences. In that series, first one was done by the Aristotle. He classified organisms, plant as well as animal. Plants he classified. Earliest attempt of classification, we can say the credit goes to Aristotle. Earliest attempt of classification, credit goes to Aristotle, a Greek philosopher. He classified plants under three categories. That is herbs, shrubs and trees. Herbs, shrubs and trees. At that time, there was no scope of microscopy. Only external features were considered by Aristotle to classify them. He classified plants under three categories, animals under two categories on the basis of blood. That is, with RBC and without RBC. With and without RBC. With or without RBC. Or we can say at that moment there is no chance to see them. So with red blood and without red blood. With red blood and without red blood. Then in modern era, that is 17th, better to say 18th century, the proper classification of organisms were started. And first one, that is given by the Mr. Linnaeus, that is he gave two kingdom system of classification. Then three kingdom system of classification was given by the Ernst Haeckel. Ernst Haeckel gave three kingdom system of classification. Four kingdom was given by Copeland, and right now which is applicable and which is studied in detail is five kingdom system of classification. Five kingdom system of classification was given by R.H. Whittaker. Five kingdom system of classification was given by R.H. Whittaker. Five kingdom system of classification, it was given by R.H. Whittaker, American taxonomist. R.H. Whittaker, he was American taxonomist. And above mentioned four kingdoms are them. That is Monera Protesta. It is actually Protesta. this A is not there. Fungi, Plantae, and one more is Animalia. The fifth kingdom which is not here in display, that is Animalia. His fifth kingdom was Animalia. And every kingdom has given a specific title. First one, Monera, kingdom of microbes. This is wrong. Why? Microbes are there, but they are all prokaryote. They are all prokaryotes. So Monera is actually kingdom of prokaryote. Then come to the second one, that is Protista. Protista kingdom is kingdom of unicellular eukaryote. Protista kingdom, that is kingdom of unicellular eukaryote. And this is a right match here. Kingdom of, what about the fungi? Kingdom of multicellular consumer, it is wrong. It is kingdom of multicellular decomposer. For fungi we can say kingdom of multicellular decomposer. And the same with the Plantae. They are not decomposer, they are all producers. They are all producers. So fungi, they are decomposer. The correct one with the fungi is decomposers. Fungi are decomposers. And Plantaes, they are actually producer. They are actually producers. That's why the correct option is second, that is Protista, is kingdom of unicellular eukaryote. Protista is also considered as link between prokaryote and multicellular eukaryote. Connecting link. We can say connecting link. Connecting link between prokaryote and multicellular eukaryote. Multicellular eukaryote. Multicellular eukaryotes. This is about Protista. Term Protista was given by the Haeckel. Term first time used by Haeckel. Term was given by the Haeckel. So this is question number 1 and its correct answer is Protista. That is correct answer in this series is second one. This is correct answer. Second question, choose correct match. 2 columns are given. 2 kingdom system, 3 kingdom system, 4 kingdom system and 5 kingdom system. 2 kingdom system Linnaeus. 2 kingdom system was given by the Linnaeus. That is with one, first one is C. 3 kingdom system was given by Haeckel. 3 kingdom, that is from second. It is already matched. 4 kingdom system Copeland. 4 kingdom system Copeland. And 5 kingdom system was given by Whittaker. So CBDA. That is first one incorrect match, second CB but AD, here DA. So that is third one is correct. Third one is correct. In this series, third one is correct. It is properly matched. 2 kingdom system was given by Linnaeus. 2 kingdom system of classification was given by the Linnaeus. That is this fellow. Then 3 kingdom Haeckel, right. 4 kingdom was given by the Copeland. 4 kingdom system was given by the Copeland. And the 5 modern system was given by the Whittaker. 5 kingdom system was given by the Whittaker. In 2 kingdom system of classes, third question now. Third question, 2 kingdom system did not differentiate between. Underline, I prefer underlining the proper, which is as a key points. here did not differentiate between that an organism with non-cellulosic cell wall and the cellulosic cell wall, right? Yes, there is no criteria of cell wall, it is only presence or absence. Organism with or without cell wall, this is a criteria. Algae and fungi, this is second one. They are kept together under plantae. They are not distinguished. Then fungi and angiosperm, they are also. So here the first one did not distinguish, then third one and the fourth one. That is, answer is A. Answer is 1, that is A, C, D. Why A, C, D? Actually, Linnaeus classified organisms under 2 kingdom. Number 1 plantae, and number 2 animalia. Number 1 plantae and number 2 animalia. These 2 kingdoms were given by the Mr. Linnaeus. Linnaeus classified all those which are with cell wall. That is, chemical nature is not considered by him. So that's why it doesn't matter whether cellulosic or non-cellulosic. If cell wall is there, they are kept under plantae. If cell wall is not there, they are kept under animalia. So that's why the first one, organism with non-cellulosic cell wall and cellulosic cell wall. His criteria is only presence or absence of cell wall. So that's why not distinguish these, that is, cell wall is the only criteria. Then algae and fungi both are with cell wall. So Linnaeus kept them under plantae. Then third one, fungi and angiosperm, they are also with the cell wall, they are also kept under plantae. Whereas nowadays fungi are totally separated from all and kept under the separate kingdom, that is kingdom fungi, and on the basis of nutrition by Mr. Whittaker. So it was a very early stage of the classification in which in-depth study of a cell is not possible. And the criterion which were used by the Linnaeus, they are also so limited. So at that moment this was correct actually. Now come to the fourth question. In 5 kingdom system, which one is not the main basis? Which one is the main basis? That is, number of criteria were used by the Mr. Whittaker. 5 kingdom system of classification was given by Mr. Whittaker. 5 kingdom system of classification was given by the Whittaker, and number of criteria were there. For example, complexity of cell. First criteria is complexity of cell. That is, on the basis of this feature he was able to distinguish prokaryote and eukaryote. Second, complexity of organism. Complexity of organisms, that is, unicellular and multicelled organisms are separated on the basis of this feature. Third, mode of nutrition. This is the main criteria of Whittaker's classification. Mode of nutrition is main criteria of Whittaker's classification, and that's why he removed fungi from plantae. On the basis of mode of nutrition, Whittaker removed fungi from plant kingdom and separated and established a new kingdom, that is kingdom fungi, on the basis of mode of nutrition. So this criteria was the main criteria of Whittaker. That's why he was able to separate he was able to separate fungi from plantae. Then other criterion ecological role, that is producer, consumers and decomposers are separated. Then reproduction is another criteria of Whittaker. And the last one is phylogeny, that is evolutionary history. Phylogeny or evolutionary history. So these are the criteria of Whittaker's classification, out of which mode of nutrition was the main criteria. Whittaker, American taxonomist, gave the 5 kingdom system of classification in 1969. 5 kingdom system of classification given by Whittaker, 1969. And his main criteria for classification was mode of nutrition. Before Whittaker, fungi were included under plant. But Whittaker's this criteria, that is mode of nutrition, separated fungi from plantae and established a new kingdom, that is kingdom fungi. Then come to the fifth one. Cell wall is non-cellulosic. Total 5 kingdoms are there. In which cell wall is present in kingdom Monera. Cell wall is seen in kingdom Monera. It is also seen in the kingdom Protista in some members, that is Protista with or without cell wall. Then fungi, they are also with cell wall. Then plantae is also with cell wall, but animalians, they don't have cell wall. All 4 are with cell wall. These are all 4 with the cell wall. But here the question is non-cellulosic. Cell wall. Monerian cell wall non-cellulosic. It is made up of mucopeptide complex. Cell wall in Monera is made up of mucopeptide complex. That is, it is non-cellulosic. So first one is Monera. Let's separate. That is non-cellulosic cell wall. The first one, that is number 1 Monera. Then Protistans, they have cellulosic cell wall plus extra material is also there. Then fungi, non-cellulosic cell wall. In fungi, cell wall is non-cellulosic. The second group, that is, they don't have cellulose in their cell wall, fungi. Plantae, they have cell wall and cellulosic. So most correct answer is Monera and fungi. Animalians, they don't have cell wall. So if we classify organisms on the basis of chemical nature of cell wall, so there are two categories. With cellulose, then only plantae is answer. Plantae and some members of Protista. And without cellulose, in that case only 2 options are there. That is, first one is Monera. Monerian cell wall is non-cellulosic, made up of mucopeptide complex. Similarly in fungi, the cell wall is non-cellulosic, made up of chitin. So in these 2 cases, cell wall is there, but it is non-cellulosic. So the correct answer is Monera and fungi. Now maximum nutritional diversity. That is the only criteria, that is nutrition. And my answer is Monera. Why? Monerians are autotroph as well as heterotroph. First one, Monarians are autotroph as well as heterotroph. That is, two options are already there on the basis of nutrition, autotroph and heterotroph. Autotrophs, those who are able to prepare food themselves, whereas heterotrophs, those who are dependent. Then again, autotrophs are further divided under two categories: photosynthetic autotroph. Photosynthetic autotroph, better to say photoautotroph. Photosynthetic autotrophs are photoautotroph, that is, they prepare food by the photosynthesis process. And chemosynthetic autotroph, that is, chemical energy of chemicals is used. So they are called chemoautotroph. This is unique feature restricted to Monera, few members of Monera. Chemoautotrophs. Then again, photoautotrophs are also of two categories. Number one, lithotrophs, that is organic electron donor is inorganic material, that is lithotrophs. Then organotrophs, that is organic material as electron donor, organotrophs. So you can see various options are there in the nutrition. Heterotrophs are also further categorized in the three categories, that is symbiont, then parasites and saprobes. So there are various types of organisms are there on the basis of nutrition. This type of nutritional diversity can't be seen in the other members. Plantae, they are autotrophs only. Fungi, they are heterotroph only. Animalians are heterotroph only. Monarians are autotroph, heterotroph. Then autotrophs are further divided as well as heterotrophs are further divided. That's why maximum nutritional diversity is seen in the member of kingdom Monera. Then our next question is absorptive heterotrophs. Algae are autotroph and they are photosynthetic autotrophs. Second, fungi. Fungi are heterotroph. Here fungi are heterotroph, that is not able to prepare their food. Then fungi release enzymes on foodstuff. Fungi release enzymes on foodstuff and make it soluble and simpler. They make it soluble and simpler. Followed by they absorb from their surface. So they absorb from their surface. Fungi release enzymes or digestive enzymes over the substrate or nutritive material. Then it is converted into simpler and soluble state. Then after they absorb. So they are absorptive and the material is from other resources. So they are heterotroph. So my answer for this question is only fungi. Algae autotroph, bryophytes are type of plants, they are also autotroph, and pteridophytes are also autotroph. So the only possible answer in this question is fungi. That is not able to prepare their food, they are heterotroph. And what kind of heterotroph? That is, they release enzymes from their cells and make the substance soluble and simpler, followed by the absorption. Now decomposers. Decomposer, that is saprotrophs. In Monera, yes, Monarians, they have decomposers also. Monarians are auto as well as hetero, and hetero further divided under three categories, that is symbiont, saprotroph are decomposers, and parasites also. First, Monarians are also decomposers. Then fungi, they are exclusively decomposers. Fungi, they are exclusively decomposer. Another one is fungi. Another one is fungi. Then in this case, the possible answer is first one Monera, second few members of Protista, and most of the fungi. Annihilants are heterotroph, but they are not decomposer, they are consumers. Annihilants are consumers, so they are not kept under this category. Annihilants are consumer. Annihilants are consumers, where the question is asked about the decomposers only. So final answer is third, that is Monera, Protista and fungi. Some members Monera, some members of Protista, bulk members of fungi. Fungi are mostly decomposer. It's a kingdom of decomposer, multicellular decomposer. Kingdom of multicellular decomposer. Then answer is fungi also. Question number 9. Monarians which are found in extreme environmental condition where the ordinary organisms can't possible. That is in normal habitat. Normal habitat is universal for all. That is, it is applicable for the most term. But in extreme condition, that is, archaebacteria are found. Archaebacteria are found in extreme environmental condition. Even they represent the early form of life. Archaebacteria represents very early form of life. Early form of life. And archaebacteria are also termed as oldest living fossil. Oldest living fossils. Archaebacteria are called oldest living fossils. Archaebacteria, those who are found in marshy area, they are methanogens. Then extreme salinity, halophiles. Then thermoacidophile, there are two adverse conditions. Number one, temperature, that is high temperature, that is 75-80 plus. Second, extremely low pH, that is hardly at 2 pH. All three types of archaebacteria. First one, methanogens are methane producing archaebacteria. They are found in the marshy area. Second, halophiles, high salinity, that is again adverse condition. Halophiles. And third one, high temperature, that's why thermo word is used. Then extremely acidic, that's why acido. Philic, that is phile, means philic. Thermoacidophile, extremely high temperature, that is 80 plus, and the pH of that particular environment, is less or 2 pH approximate. So very strong acidic condition is there. So only archaebacteria, they are found in extreme environmental conditions. Eubacteria, normal conditions. Cyanobacteria, that are blue-green algae. They are blue-green algae, they are found in aquatic habitat. And mycoplasma, free living as well as parasite. Mostly parasite.